Hey everyone, I hope you all are doing well. So, welcome back to Windows Seat Studies. So, till now we have learned about different methods for detection of 5-methyl cytosine and 5-hydroxymethyl cytosine. So, we have learned about bisulfite sequencing, oxidative bisulfite sequencing, and tap sequencing. So, let's do a quick recap of what those sequencing method do. So, in bisulfite sequencing, the sodium bisulfite deaminates the cytosine, converts it into the uracil, and that is read as the thymine in the sequencing methods. So cytosine is converted to thymine in bisulfite sequencing, while 5-methyl cytosine and 5-hydroxymethyl cytosine stays as it is, and that is why they read as cytosine in the sequencing method. In the oxidative bisulfite sequencing, we add one more step, and that is oxidation of 5-hydroxymethyl, and the oxidation of 5-hydroxymethyl results into 5-formyl cytosine, and this 5-formyl cytosine can be deaminated by sodium bisulfite, and this 5-formyl cytosine gets converted into uracil and it is read as thymine. Okay, so while 5-methyl cytosine, it stays as it is not deaminated, and that is why it is read as cytosine. So this was oxidative bisulfite sequencing. So in the bisulfite sequencing, we can detect 5-methyl cytosine, but we cannot differentiate between 5-methyl cytosine and 5-hydroxymethyl cytosine. But in oxidative bisulfite sequencing, we can detect 5-hydroxymethyl cytosine and 5-methyl cytosine, means we can differentiate between both of them. While tab sequencing is a different method, this uses the enzymes as TET, as well as it also uses beta GT, beta glucosyl transferase, which transfers the glucose group on the 5-hydroxymethyl cytosine, and that is why it gets converted into 5-glucosyl hydroxymethyl cytosine. So this 5-GMC, this conversion of hydroxymethyl cytosine to 5-GMC, this protects the 5-hydroxymethyl cytosine, and so that TET enzyme cannot work on it. So TET enzymes work on 5-methyl cytosine, and it converts, it converts it into 5-formyl cytosine and and then further into 5-carboxyl cytosine, and on this 5-carboxyl cytosine, the sodium bisulfite can work, and that is why it is deaminated. That is why in the tap sequencing, along with the cytosine, 5-hydroxymethyl cytosine is also deaminated. Why? Because this is already converted into 5-carboxyl cytosine by TET enzymes. So this was all about the different methods, and that is why uh, we are going to solve a question related to those sequencing method, that how do we differentiate between those. So this is a question here. This is the original sequence of DNA. This is the real sequence of DNA. Okay, and the objective of this question is that you have to tell whether the DNA is unmethylated, means on which CpG dinucleotide it is unmethylated, means it is cytosine, where it is methylated, means it is 5-methyl cytosine, and where it is dimethylated, or you can say 5-hydroxy methyl cytosine. So you have to identify the cytosines, 5-methyl cytosines and 5-hydroxymethyl cytosines in this DNA sequence. Okay, so this is the DNA sequence. So we have learned that in the DNA sequence, only the CpG dinucleotides get methylated. Okay, and that is why we have to identify which cytosine is cytosine, 5-methyl cytosine, and which is 5-hydroxymethyl cytosine. So first you will identify the CpG dinucleotides. Okay, so this is the CpG dinucleotide. This is the second CpG dinucleotide, and this is the third one, and this is the fourth one. So identify them in the other sequences. This is the bisulfite sequence, means the sequence that you have, the result that you have get after bisulfite sequencing, and this is the result of tap sequencing. So this is the CpG dinucleotide here. This is the second one, this is the third one, and this is the fourth one. So here this should be the CpG dinucleotide. Here this should be the one. Here this is the CpG dinucleotide, and here this should be the one. So now you have to check which is by doing bisulfite sequencing or tap sequencing, which cytosine gets deaminated and which does not. Okay, so let's see. So here in the first in the first CpG dinucleotide, the C does not get deaminated; it stays as it is. Okay, while in tap sequencing also it stays as it is. So this cytosine stays as it is in both bisulfite sequencing and tap sequencing. So let's go on to this table and let's see where it stays same in both of the cases. So the 5-hydroxymethyl cytosine, here you can see it stays deaminated in both bisulfite sequencing as well as tap sequencing, and that is why this cytosine is 5-hydroxymethyl cytosine. Okay. So now let's still take a look at the second CPG dinucleotide. Here you can see the original C gets it stays deaminated. So it stays deaminated in the bisulfite sequencing. So it can be either 5-methyl cytosine or 5-hydroxymethyl cytosine. But it gets converted into, it gets deaminated. So which one gets deaminated in tap sequencing? The 5-hydroxymethyl cytosine gets deaminated. That is why this cytosine is 5-methyl cytosine. So this is the methylated cytosine. Let's go on to the third CPG dinucleotide. Here you can see it gets it doesn't get deaminated in bisulfite sequencing, but it gets deaminated in tap sequencing. So this is also a 5-methyl cytosine. Okay, and here you can see the last one, the last cytosine in the CPG dinucleotide. Here it does not get deaminated, and even in the tap sequencing it does not get deaminated. This means this is 5-hydroxy methyl cytosine. So now you can, so now the answer would be, so you have to tell which are the hydroxymethyl cytosines, which are the 5-methyl cytosine, and which are the 5-hydroxymethyl cytosine. So you have to tell this, right? So the answer would be, the first is T. The second cytosine is actually 5-hydroxymethyl cytosine. Okay, wait. Let's do one thing. Let's write it here only. So this is first will be thymine. Second is 5-hydroxymethyl cytosine. This first one is 5-hydroxymethyl cytosine, and the second one is 5-methyl cytosine, and those will be cytosines. Okay, and this, because it won't be get methylated, right? Because it is not CPG dinucleotide. And now this CP in the this cytosine will be 5-methyl cytosine. We have deduced this cytosine will be cytosine because again it is not CPG dinucleotide, and this cytosine will be 5-hydroxymethyl cytosine. So this is how you can predict which cytosine is cytosine, while which gets methylated and converted into 5-methyl cytosine, and which is 5-hydroxymethyl cytosine. So that's it for today. I hope you, from whatever we have learned till now, you can solve the questions of this kind. Even if you have oxidative bisulfite sequencing result instead of tap sequencing, all you have to do is that make a table and predict what will cytosine as well as 5-methyl cytosine and 5-hydroxymethyl cytosine will get converted it, and you can solve the questions of this kind very easily. And that's it for today. In the next video lecture, we are going to start a very important topic, and that is histone modifications. Till then, see you. Bye bye.